Well, I want to thank you for coming out on a Saturday morning. That's pretty brave. And those of you that are here, I hope are ready for a science lesson, because that's what this is about. I love it when we do these kinds of things in a neutral setting like this, because it opens the door for folks to come visit us. And if you're here as a guest, we thank you so much for joining us. Our goal today is to talk about the great designer, evidence from the chemistry of living things. I taught chemistry for about 20 years at the college level, 4 years at the high school level, and majored in chemistry education in my degree programs, including a year at Harvard University in 67, 68. I'm telling you that to say this is in the midst of the things I'm going to be talking to you about. I was in the midst of some of the great changes that were taking place. So let me start this morning by talking a little history for those of us who are 80 years old and over. Anybody else over here in this room over 80? No. Okay, I'm the oldest guy in this room. I've lived through one of the most amazing times in the history of mankind. In the 1950s, class, and that's how I'm going to refer to you. You're my class today, so you're mine. And I expect your rapt attention. In the 1950s, things broke loose in terms of understanding life at the biochemical level. So, for example, in 1953 is when Crick and Watson discovered the structure of DNA. They received the Nobel Prize for that in 1962. We'll be talking about that a little bit. In that same decade, Friedrich Sanger sequenced insulin. That's the first protein that was ever sequenced. And you'll know what that means in a little bit. But that was 1951-52. He received the Nobel Prize for that in 58. And he's one of the few people in the history of mankind that got the Nobel Prize twice in chemistry. I don't think there's anybody else. But in the 70s, he sequenced the first DNA and received the Nobel Prize for that in 1980. All of these things, ladies and gentlemen, were opening up the world of the microscopic about life. There was another grand event that happened on October the 9th, 1957, or 50, let's see here, yeah. October the 4th, 1957. Now, some of you are old enough for that one. Who remembers, and by the way, this is a class, so you can answer my questions. 1957, October the 4th. Anybody remember what happened on that day that scared us all to death? I'll give you a hint. It has to do with the outer space. Yes. What was Sputnik? Yeah, it was a satellite, first satellite put into space by human beings. October the 4th, 1957. Scared us to death because the Russians did it, and we knew they were going to take over space and rule the world. So what happened out of that class is our government started dumping a lot of money into science education. We were behind, and we wanted to catch up. And I'll have to, if I may be a little personal, I was a personal beneficiary of that. Because one of the things they did was they gave Harvard University a bunch of money to train science teachers in a better way. So they led a program to rewrite the curricula for every high school course in science. So some of you may remember BSSC biology, Chem Study chemistry, PSSC physics, and the new math. You remember this? Not too many of you, you're too young. But it was a complete rewrite of science and math curricula. I was in the middle of that. So I'm telling you that to say I've been in the midst of everything I'm going to be talking to you about. And in some cases on the front lines, because I had a part in the Chem Study chemistry and taught it for the first time at St. Pete High many, many years ago. Important things happening because we were uncovering how life works at the chemical level. And did we beat the Russians to space? At least, did we beat them to the moon? We did. Now, how about July the 16th through the 24th, 1969? I'm going to name them. Neil Armstrong, Buzz Aldrin, and Michael Collins. What happened? They landed on the moon for the very first time, 1969. You remember our President Kennedy said, before this decade is out, we will be on the moon. It was an exciting time, class. And it was a very heady time for science. Because many people began to say, as mankind opens up all these mysteries of life, it won't be long before we understand it all, and we don't need God. That's a sad thing, but that's what began to happen. And so, this magazine article came out on April the 8th, 1966. And the headline was, "Is God Dead?" And the reason it did, class, is because that was the way things were going. And I can tell you, as a college student in those days, in a public university, I was being challenged a great deal because I'm a believer in God, was then and still am. But I was being challenged by my science professors that you better watch out about that, because there's going to come a time when you don't need God. We're going to understand how things work, and that God was not needed for these things. And that's challenging. And that is partly why I'm doing what I'm doing today. Because there are lots of people today that will tell you, if you're a believer in God, you belong in the Middle Ages. You're out of date. And I'm here to tell you, you're more up to date than anybody. There's more reason than ever to believe in God. And that's what I want to talk to you about. Well, there was a song written about then. Many of you probably know this song, "Our God, He is Alive." Have you sung that song? Well, that was our dean at the college where I sat in his seat, literally. And he was a physicist and well respected in this world. And he wrote the song in direct response to that. So we're not going to sing that song, but it's a good song, because in the chorus it says, "Our God, He is alive, in Him we live and we survive." Great song. But here's what was in that article, among other things. Thomas J.J. Altizer and William Hamilton, in their book, Radical Theology and the Death of God. Class, please understand, these two gentlemen are theologians. They're preachers. And here's what they said. We must realize that the death of God is an historical event, that God has died in our cosmos, in our history, and the dead God is the God of the historic Christian church and beyond the church of Christendom at large. So did you know that your God died in the 1960s? That's what these preachers were telling people. So it was not only coming from scientists, it was coming from theologians. So it was a time when belief in God was under great duress and stress. I like this book better. Is Atheism Dead? It's a much more current book. It's by Eric Metaxas from this century. It was published in 2020, I think. I highly recommend this to you. Eric Metaxas is not a scientist, but he's a researcher. He's a good researcher, and he's done his work very well. Copyright 2021, as you can see there. And here's what he says. Since that Time article in 1966, roughly 5 things have arisen to challenge, and I will argue to overturn, the secular consensus that formed in the wake of that article. Let me say something briefly about that. It is absolutely true that after that Time magazine, there was a secular consensus. That means no God. We don't need God. And it was very powerful. And young people, I'm talking to you because you're being told by lots of people today, you don't need God. Science will explain it all. And they're just as wrong as they can be, that they have a lot of voice. So I'm going to be talking loud today. And I hope I get my point across, that you need to hear the other side of that message. So atheism has become very strong, but it's got its problems today. And he says there are roughly 5 things that have happened to challenge what they were saying back then. And here's his 5. He says, we have learned that there's a beginning. Let me see if I can make this thing work. There you go. A beginning. Did we talk about this last night, class? Just briefly? I'm not taking that as my theme during this series of lectures, but there's books about it in my bibliography if you want to study it. The evidence from science class is that matter, space, and time had a beginning. That speaks to someone started this thing. It speaks to there must have been a God. And it's much more reasonable to say there was a mind that created matter, space, and time, rather than saying space and time somehow created itself and then we made up God, which is what the other side believes. Secondly, fine-tuning. We talked about that last night, didn't we? So I'm not going to rehearse that whole thing again, except to build on it. Everywhere you look, it sure looks like things have been designed for us to be here. And everybody admits that. Well, why don't you admit what goes with it? There must have been a fine-tuner to help make that happen. The third thing that he mentions is stupefying complexity. I like that word. I want you stupefied when I get through with you today. I want you blown away by the evidences that we've uncovered. So that's what we're going to deal with today. Then he said there's been archaeological discoveries, and that supports the text of this book. I'm not talking about that in this lecture series. But there have been a lot of fascinating archaeological discoveries. Get this book and read about it. And then finally, we've learned in this century what atheism is and what it produces. So I'm going to spend just a little time here, if you'll bear with me, and read you a little bit of what he says about what atheism has produced in the 20th century. If you hadn't thought about it, the 20th century was the century of atheist regimes. The Nazis and the communists in the 20th century. The new atheists ignore the following. It remains an unendurably horrible fact that dedicatedly atheist regimes in the last century tortured and murdered multiplied millions, usually for their Jewish or Christian faith. In fact, those murdered by the handful of atheist regimes amount to well over 150 million human beings. That's what atheism has done for us. The idea that 150 million human beings were murdered by atheist regimes might be the single most disturbing fact in the history of the human species. There has never been that level of slaughter. I lived in Romania for 2 years, class, a formerly communist country. And I can tell you that the results of the communist regime is horrendous everywhere they've touched it. You want nothing to do with it. A little more detail. In the year 1937 alone, dedicated atheistic Soviets, you hear a lot about the Nazis, right? Who murdered all the Jews that they could. The Soviets murdered way more than the Nazis. In 1937 alone, the Soviets murdered 106,300 clergymen. They wanted to wipe out religion. Marx considered it the opiate of the people. The Soviets, in their course of their adoption of what they call scientific atheism, murdered between 12 and 20 million human beings, just them alone. And they did it partly on the basis of science, which led them to say there is no God, so we can do what we think is right. Well, there's a whole lot more about that in this book that you need to look into. We don't want atheism, and I will tell you, if you don't appreciate the United States of America and the freedoms we have here, you need to go live in a communist country for a while. I would say that to anyone who openly belittles what's going on here in our country. We have more freedoms than any system has ever had in the history of mankind, including what we're doing right now. So, is atheism dead? I hope it's on the way. It's a worthwhile book. So I'm going to do what I did yesterday. I'm going to refer to one passage of Scripture in this book, which is the book for Christianity, isn't it? Romans 1 verse 20 says, I'll just read it to you. For since the creation of the world, his invisible attributes are clearly seen, being understood by the things that are made, even his eternal power and Godhead, so that they are without excuse. Because although they knew God, they did not glorify him as God, nor were thankful, but became futile in their thoughts, and their foolish hearts were darkened. This passage teaches you can know that he's a powerful being that's not part of this nature. He's outside of nature. And it's the argument from design. The Bible itself says nature teaches you there's a God. Without this book. So in this series of lessons, I'm not talking about the Bible. I'm talking about God's other book, which is also very clear in its testimony that there is a God. So class, that's my introduction this morning. Welcome. But you didn't come for that. You came for this. So we begin now with our chemistry lesson for today. I know you're excited. Are you excited? That sounds very weak to me. Are you excited? Yes. Oh, good. Well, I certainly am. Because if there's anything more exciting or almost as exciting as teaching from this book, it's teaching you from this other book. And I always felt when teaching chemistry and mathematics, that's what I was doing. Teaching God's thoughts after him. So welcome. We're going to talk about the great designer. So to do that, we need to look at how life works. So I thought I would use a book by Dr. Francis Crick. I mentioned that name already once this morning. Francis Crick. Can somebody help me? What's he famous for? He helped discover what? The structure of DNA in 1953. He and James Watson. He's an amazing chemist. So I have nothing but honor for him in terms of what he discovered. And folks, everybody knows about DNA now. Prior to 1953, nobody knew about it. So in the last 75 years, we've uncovered one of the key secrets of life. So he wrote this book in the 1980s, 1981, and it's called Life Itself, Its Origin and Nature. So let's listen to Francis Crick. If there's anybody that could tell us how life works, it's Dr. Crick. So I'm turning to page 72, and here we go. Life as we know it on Earth appears as a synthesis of two macromolecular systems. And I've just lost you, haven't I? Let me make it a little simpler. Every living thing has two types of giant molecules. And I'm going to explain that to you. That's what he's saying. Every life form we know about has two kinds of molecules. Here we go. The proteins, because of their versatility and chemical reactivity, do all the work, but are unable to reproduce themselves in any simple way. So let me introduce you, class, first to the proteins. In life, there are critical components. The structural materials, that is, those things that make up the shape and mobility of a thing, are made up of proteins. And proteins are assembled amino acids. So, class, I need you to say that with me, all right? So let's read it together. Proteins are assembled from amino acids. A protein is a string of amino acids. I'm going to explain that to you more fully in just a minute. The proteins make up the structural materials of living things. Secondly, there's the tools and machinery. That's the metabolism, growth and maintenance. Those are enzymes. But class, an enzyme is just a special case of a protein. It also is assembled from amino acids. So these first two are both proteins. So class, last night I gave you the answer to the questions for most of my questions. Remember, it was just right. Today, the answer to most of my questions is protein. So let's practice. Most of life depends on protein. Good. So when I ask you a question, most of the time the answer is going to be proteins. So you're ready to do that, right? And don't be shy. I'd expect enthusiastic answers. Proteins are a big deal. Every living thing, from one-celled organisms to up to you, has proteins and lots of them. But there's another one, and that is the blueprints, or the things that allow reproduction. These are the molecules that contain information and that direct your whole body. And they are what we call the nucleic acids, or DNA and RNA. Every living thing has both. Can't have a living thing without both. Here's what Dr. Crick says about that. The problem of the origin of life. So how did life get started, class? That's the biggest question in the universe. How did we get here? The origin of life would be a great deal easier to approach if there was only one family of molecules capable of doing both jobs. But life as we know it employs always two. And that means you must have them both. And he says this may well be due to the fact that no macromolecule exists which could conveniently carry out both functions. Well, it's been 60 years since he discovered all these things. And have we found, class, any living thing that doesn't have to have these? No, we have not. You have to have them both. That is a huge problem for an atheist. Because to get life functioning, you have to have both. And when I get through with you today, I hope you'll have a deeper appreciation for how important both are. Now, I'm going to read you one other thing he says here, and it's a little bit dangerous. But I want you to understand, I did not say this. He says, RNA and DNA are the dumb blondes of the biomolecular world, capable for reproduction with a little help from proteins, but of little use for very, really demanding work. I'm sorry, ladies, but that's probably the only thing you'll remember from my speech today. But the fact is, RNA and DNA allow for reproduction. Proteins do not. But class, you can't reproduce anything without proteins. You have to have them both. And they're critically important. And the more we learn, the more critically important it gets to have both of them functioning. So, welcome to Biochemistry 101. I'm so glad to have you in my class today. And this is where we begin class, because this is only one of the most significant things I'm going to say to you the entire semester. So you better stick with me. When you come back to class tomorrow, there will be a quiz, and it'll have this on it. Because every protein and every living thing is made up of these 20 amino acids. And did you hear what I just said? In single-celled organisms, these are the amino acids. In you, the same amino acids. Doesn't matter what kind of living thing it is, the proteins in it are made up of these. That's kind of astounding, isn't it? Do you know what an evolutionist says about that, class? An evolutionist says the reason we have a common chemistry in all living things is because if you trace your ancestry back, we all are kin to each other in one tree of life. And the first organisms, way back down there, little single-celled organisms, evolved from non-living matter by itself, and out of them came all the other living things. That's why we have a common chemistry. Class, that is not a foolish answer. That is a possible answer to this phenomenon. It is the truth that all living things have a common chemistry. So don't argue with facts. What does a believer in God say about that? Here's what I say. It's perfectly reasonable that if a creator was going to produce a living system on which we depend on one another, that we have a common chemistry. You don't have to start over every time you make something, do you? No, in fact, that's a foolish thing to do. A common chemistry means I can feed on those little organisms, and they can feed on their other little organisms, and we can build a system that works. Look, you're not going to answer that question from just this information. But the fact is there are two possible answers to this. They were either designed that way or they came about naturally that way by an evolutionary process. You with me so far? But it is a fact, class. Glycine, alanine, valine, leucine, isoleucine, proline, serine, threonine. You need to learn how to pronounce these things. And tomorrow in our first quiz, I want you to be able to name them all and their three-letter abbreviation. That's not too much. Most of the three letters are just the first three in the word, aren't they? Can you see that? Let me see this pointer here. You see, alanine is ALA. That's the first three letters. Valine is VAL. That's the first three letters. And so on. Some of them aren't that way, but most of them are. Maybe all of them are. No, right here, this one isn't. Cysteine is CYSH. But you need to learn those if you want to start here. And why is that? Here's why. Every protein is a combination of these 20. I don't care where you take it. For any living thing, they're made up of these 20. It's very much like in the English language. We have 26 letters in our alphabet. Any word in English is a combination of those 26 letters, isn't it? Even anti-disestablishmentarianism. That's still a combination of the 26 letters. Same with this. This is like a alphabet for proteins. Are you with me, class? Make me feel better. Okay, good. All right, so that's why this is so important. So I'm not going to do this to you, but the next thing you would learn is the structure. Now, I'm not going to make you memorize all the structure, but you at least need to know, why do they call them amino acids? Well, look, let's go back. Get rid of that for just a minute. And let's look at this. Do you see that little NH3 right there? Do you see an NH3 there? And one there. Do you see that every one of them has an NH3 group? An NH3 group is called an amine. So that's an amino. And so why does it call it an acid? Well, look at that thing. Where did I go? Here we go. You see the COO right there? Can you find one of those in every one of them? Look through there. There's a COO on that one too, isn't there? COO is an organic acid because it releases a hydrogen ion, and that's what makes an acid. So they're called amino acids, and every one of them has that same structure. But do you notice they're all different also? They have slightly different structures. Some of them significantly different. Every one of them has an amine and an acid, but they're all different in terms of their... But do you see what's in most of them? There's a nitrogen, and what else? Help me.
Helium.
No, not helium, it's hydrogen. And then what's the C? Carbon. And do you see anything else in there besides carbon, hydrogen, and oxygen? Did you say oxygen? Okay. And nitrogen, right? Anybody see anything else? There is one other thing. Yeah, right here. That's sulfur. And there's a sulfur down there also. So class, living things in terms of their common chemistry have basically 5 different elements involved. That's all. Isn't that amazing? So they have a lot of commonality. And I'm sure there was a design behind that. I mean, it makes sense to me. All right, but that's the beginning. So what I will want you to do is learn some of these structures, because why is that important? When you string a bunch of amino acids together, the order in which they're strung and the structure of their chemical structure is critically important to what they can do. Listen to me, class. Every protein has unique things that it can do and nobody else can do it. So while you're sitting there, you've got thousands of proteins doing different jobs, and they are required for the jobs they're doing. And if you mess up and make a different protein, it doesn't work. Okay. I apologize to every protein for the incomplete treatment I have just given it. We should spend the next 6 weeks on proteins. I don't have time to do that. I got to talk about the other major molecule, that's DNA. And let's talk a little bit about it. It is a double helix with crossbars. Let's see if I can point to that. Do you see there's a strand right here? Well, it's hard to guide this thing. See that strand? And then here's another strand. And can you see they're wrapped around each other? That's what Crick and Watson discovered in 1953, is an interactive couple of strands. And do you see there's cross pieces here? Cross pieces? And in this picture, there are exactly 4 colors. I'm going to use my stick for that. I'm tired of messing with that thing. There's a green and a blue, a red and a yellow. Everybody with me? That means there's exactly 4 bases that provide the cross pieces. The green always goes with the blue, the red always goes with the yellow. They're not that color class. This is a model. But that does help you remember, doesn't it? So what did I do with my thingy? Here it is. This gives it the capacity to reproduce itself. It contains a language with 4 letters. What Crick and Watson uncovered in the 1950s, by the 60s and early 70s, they had figured out has a language in it. An incredible language with 4 letters. And every word in that language has 3 letters in it. It's a language with only 3-letter words. Kids, wouldn't that be nice if every word was just 3 letters? Like and but and sit. What else? Give me another one. The. In this language, every word is 3 letters long. And you only have 4 letters to use. You can repeat. So watch. Here's the whole language right here in front of you. It's called the genetic code. And if you take 4 letters, and you can use them 3 at a time with repeats, how many words can you make? Anybody know? That's a math problem. Well, let me tell you how to figure it out. In the first position, how many choices do you have for letters? 4. In the second place, how many choices do you have? No, you can repeat. Well, let's wait till the child gets out. Thanks for bringing your daughter, by the way. Now let's start over. You have 3-letter words. You're in the first position. How many letters can you put in that position? All 4 of them. How about in the second position? All 4 of them, because you can repeat. And the third? All 4. So there are 4 times 4 times 4 possibilities. So do the math for me. Come on. Kids, what's 4 times 4? 16, and 4 times 16 is? You're not a kid. But you could do it, couldn't you, kids? 4 times 16, can you do it in your head? 64, yeah. So class, there's exactly 64 words up here. Let me show you how to read. May I teach you how to read? All right, I'm going to use my pointer again. If the first letter is T, that's thymine. Well, let's just say there's thymine, cytosine, adenine, and guanine. Those are the 4 letters. They're 4 chemicals. And guess what? Next week there will be a quiz on that. You need to know them by name. Thymine, cytosine, adenine, and guanine. And by the way, A always goes with T and C goes with G. They match up because of their chemistry class. But I'm teaching you how to read. Suppose the first letter is T, and the second letter is T, and the third letter is T, you get this word, T-T-T. It's pronounced t-t-t. This is not a spoken language, ladies and gentlemen. But T-T-T is a word, and here's what it means, P-H-E. Now that's really helpful, isn't it? So if I were to pronounce it for you, t-t-t means fee. Does anybody know what fee means? That's because you didn't memorize what I told you to just a while ago. You haven't had time to memorize it yet. But PHE is an abbreviation for phenylalanine, which is one of the amino acids, which is what you use to build protein. That was weak, class. Let me say that again. Phenylalanine is one of the amino acids used to build proteins. That's correct. So this code right here, TTT, stands for the amino acid phenylalanine. One of those 20. Interesting. Let's try another one. What if your word is C and A and T? That's an easy one. Right here. That's cat. And it means H-I-S. Anybody remember that one? That's histidine. That's another one of the amino acids. So the code cat means histidine. And you keep learning this and you'll find that on this chart is a code for all 20 amino acids. In fact, there's 64 words up here and there's only 20 amino acids. So let me get rid of that. Do you see some repeats up here? Yeah. There's 2 for phenylalanine right here. There's what, 6 for leucine. Isoleucine, there's 3. Here's methionine. Here's valine, there's 4 right there. So there's repeats. I call that a backup system, class. You have more than one code for the 20 amino acids. But the fact is, there's a code built into your living system that has a language that we know what it means and we know what it's doing. Now, if you were a jury in a court of law and I was trying to convince you somebody designed this, it didn't just come about by natural causes, here's the first thing I'd say. Class, there has never in the history of mankind ever been discovered anything that had a language in it that didn't come from intelligence. Nature doesn't make up languages. You listening to me, jury? That's the first thing. And every living thing has a language in it. Somebody designed that. There has never been a language in any culture, anywhere, that wasn't designed by an intelligent source. Period. If you think there is, bring it to me. I'm looking. Everybody knows there isn't any such thing. So you have to believe, if you're an atheist, that nature somehow made up a language by itself. But that's hardly beginning. That doesn't touch the hem of the garment, class. That's just the first little tiny, teensy piece. It's important. But there's so much more going on in there that's being done with that language. So let me give you a brief summary here. Just a brief summary. This is the inside of a nucleus for a cell, a eukaryotic cell, and the DNA is mostly inside the nucleus. Here's the DNA up here. This is where all the information is stored. That has the language in it. You have to copy that information. That's called transcription. Because if the information just stays in the DNA, nothing happens. You have to copy it. That's called transcription. I'm going to tell you a little bit about that. Secondly, once it's transcribed, it has to be acted upon here. Here's an example. You take messenger RNA, that's copied off of the DNA. Ribosomal RNA is copied off the DNA. Transfer RNA is copied. So all of these are copied. And there's a bunch of other things that happen that I'm not talking to you about. Please understand. We're going to focus on this one. Once it's copied, can you see here that it's got to be edited? There's an editing process. And class, let me just back up. What do you think it takes to allow you to copy DNA? Proteins. That was weak, but that's the answer I'm telling you. May I say it again? What do you think it takes to make a copy of the information on the DNA? Proteins. Only it's plural. Say it again. Proteins. It takes proteins to make this happen. Second, once it's made and you got to edit this, guess what it takes, class? Proteins to edit the copy. See these little white things? Those are called introns. They are edited out and then it's reorganized. It's spliced. What do you think it takes to splice it back together? Proteins. In fact, very complex, incredibly challenging proteins. So you got proteins here, proteins here, proteins here. Then it has to migrate. That means it has to take this information and go outside the nucleus through that hole right there. Every nuclear membrane has holes in it. Class, do you think that any respectable nuclear membrane is going to let just anything go through those holes? No. What do you think guards the hole, class? Protein. That was weak. Where are you? What do you think guards the hole? Protein. On both sides. So when this mature messenger RNA migrates up here to this hole, a protein says, well, come on through. And he opens up, lets the messenger RNA through, and then closes back up so nothing else can get out. What do you think on the other side? I must have hit something. Let's go back. I want this one still. What do you think happens on the other side of that hole? What do you think guards that side? Protein. They don't want anything going in there either that's not allowed. There's only certain things allowed in and out of there, guarded by protein. Thank you. So here comes the messenger RNA. It's now outside the nuclear membrane, and it is picked up by two ducks hugging each other. Isn't that what that looks like? That is not what it looks like, class. This is a picture to give you some idea of what's going on. But I have to say it looks like two ducks hugging each other. At any rate, the messenger RNA is picked up and you read the information off of it like a tape through a tape player. Fascinating stuff. These things are called ribosomes. Critically important. Guess what ribosomes are made of, class? Protein. Yes, and a bunch of other stuff. There's ribosomal RNA that I also mentioned up here, but there's also a bunch of protein in every ribosome. Got it? Now, class, what do you think it takes to be able to read the information off of this and translate it so that you can build a protein? What do you think it takes at this step right here? Protein. Amen. So when this thing is being read, it'll see a three-letter code. You see over here? G-U-G. GUG. That's one of those three-letter words. It means an amino acid. And so this guy will say, get me that particular amino acid. So over here in the cytoplasm is a bunch of transfer RNA. There are exactly 20 transfer RNAs for the 20 amino acids. You listening? 20 specific transfer RNAs, one for each amino acid. And going with them, there's a specific, what do you think, for each transfer RNA? Protein. Yes, that's 20 more proteins, class. Has somebody been keeping track here? No. Have we said protein for every step of this way? Yeah, and by the way, in every case, it's multiple proteins, not just one. So for every one of these transfer RNAs, there's another protein that has to go with it when they bring it over here to introduce it inside this ribosome to allow it to build the protein. And then at the bottom, out comes the protein, which is a string of amino acids, methionine, glycine, isoleucine, valine, glutamine, cysteine, and so on. That string of amino acids is a protein. So you've just built one protein. Class, to build one protein requires at least 50 different proteins. You didn't get that. I'm going to say it again. You listen to me. To build one protein requires at least 50 different proteins, or you'll never get it. Here's the big problem. At the very beginning here, it takes a protein to allow that to unzip. But class, the protein needed to make that unzip has to be built also by the same process. Are you getting me? So how'd you get that first protein when there wasn't one to even unzip the DNA yet? It's impossible. It's a big problem for atheists. That's why Crick says it may be, this is a real challenge. And let me just go a side note here. Don't lose where you are. But I have to tell you this. In this same book, this Nobel Prize winning scientist, in his little book, Life Itself, spends the last 4 chapters of this book telling you that life might have been sent here on a spaceship. Did you get what I just said? He says on page 88, an honest man armed with all the knowledge available to us now, which was 1980. We didn't know hardly doodly squat in 1980. We have learned so much more since 1980, and the picture is 10,000 times worse than it was in 1980. For how did the first living thing get started? He says in 1980, an honest man armed with all the knowledge available to us now could only state, in some sense, the origin of life appears at the moment to be almost a miracle. Reminds me of the biblical statement that one man made, I'm almost persuaded to be a Christian. Are you kidding me? Almost this needed to be a miracle. So many are the conditions which would have had to have been satisfied to get it going. No, sir. So many are the necessities and the complexities that we don't have a clue how that could happen without a designer. But this man will not have a designer, I'm telling you. I have met a lot of these folks in my life, and I've found no more hard-headed, closed-minded people. They will not let God's foot in the door. So they will take any explanation other than God. So this man, Nobel Prize laureate, spends a whole chapter telling what the spaceship might have been like that was sent here with the first simple living things. Class, is there one scintilla of scientific evidence that somebody sent the first life free on a spaceship? There isn't anything like that anywhere in anybody's ever known. It's a pure fiction. But he'll take that over God. Say it again. Yeah, he tells you what it might have been like. And he has a fancy name for his theory. It's called directed panspermia, which means they sent sperms from somewhere else. It's a fairy tale written by a Nobel laureate in this book. You read it for yourself. It's incredible. Folks, this was designed. It's better than any factory man has produced. You see why I get upset? And then they talk about God believers are foolish, ought to belong to the Middle Ages. This is the greatest fanciful tale in the history of mankind. All right, what I want to do next is I want to show you a video that kind of brings this to life. So, Troy, are you ready? This is where I'm putting my man to the test. Because this video has to stop and start at my command, and who am I commanding? And if he doesn't do it exactly right, he's going to mess up my entire lecture. No pressure. No, Troy's been working on this and practicing, so he's gotten good at it. But it's not been simple, class, because we've had to mess around, and not only do we have stupid pointers like that you can't control, but this pointer interfered with his computer for part of the time. So I think he's got that worked out. We're going to find out, aren't we? All right, I skipped a slide there, didn't I? There you go, thank you. Drew Berry, class, I want to introduce you to him. He is a biomedical animator. Kids, y'all know what an animator is? That's a guy that makes videos that picture what's happening. And he does amazing stuff. Because down at the molecular level, you can't just look at it. You have to imagine it with the chemistry that goes with it, and then he makes pictures for you. So he is a biomedical animator, works for the Medical Research Institute in Melbourne. And if you want to see something beautiful, you go to his website and look at some of the things he's done. I'm going to use two of them today. So here's one. Here we go. This is a Drew Berry animation of building a protein. Okay? And I don't think I'm going to trust this. We're going to go back to the old-fashioned way. All right, stop right there. This is the start of a gene for a bone strength protein. We're going to show you how a bone strength protein is being built. One of thousands in your body. Here's the DNA strand. Can you see how it's wrapped around itself? And here is a glob of, what do you think, class? Protein. Yes, that rides along the DNA strand down inside your cells. Remember, this is down in the nucleus of your cells. And this strand of proteins is what allows you to build a protein. So let's look at the colors here. By the way, they're not this color, class. This one is not red, but he colors it that way, so it's different from this one, which is different from this one, and this one, and that one, and that one. That's 1, 2, 3, 4, 5. That's 6 different proteins, all needed to make this happen. So let's go. We're going to watch it a little bit. And is it having trouble? Stop right there. See vitamin D? Whoops, we missed it. Go a little bit further. Is it jerking? See, it's giving him fits. Is that what it is? Oh boy. Are you going to be able to survive? Emerson, help me out here, guy. You see what I mean by electronics? They're nice, but if they mess up, you're in trouble. So what are we doing? Go back to the beginning?
He's going to plug in his laptop, so it's hardware.
Okay. While he's doing that, class, let's not waste time here. You've already seen 6 proteins, right? As we go through this process, we're going to be adding more and more proteins to show you what it takes to make one protein. Remember, we're building one protein, and the strand of DNA that I told you about is where all the information is. That's where the language is. So you should know, if you don't, that all we're talking about today is building proteins. That is not all that's happening inside your body. It's all kinds of things going on in addition to this at the same time. And nothing runs into each other. And this is all down at the nano level, class. It's amazing. All right, so what do you think? You're going to work from over there. Okay, then I'll get over here. Let's go on to where we're about to see the vitamin D. Here we go. Stop right there. All right, you see the vitamin D here? In your cytoplasm, you need vitamin D. Vitamin D, kids. Quit staying inside looking at your computer all day long. Get out in the sun. You need vitamin D. Why do you need vitamin D? Because vitamin D activates this whole process. Watch. Here comes the vitamin D. And it's grabbed by this, stop. It's grabbed by this red protein, which turns green. No, it does not. So why does he do that? Because red means stop, green means go. So that's why he does it. But all it means is this vitamin D activated that protein to start this entire process. With me? Okay, so the protein activation is now going to allow more things to happen. So here we go, Troy. So you see another glob of, what do you think, class? Yeah, stop right there. Here's another 1, 2, 3 proteins that have been added to this glob to make this process begin. And each one of them has their own function. And if you were in my biochemistry class, ma'am, you would have an assignment. I would assign you the green guy to come back to the class and tell us what it does. And you would have one of the other colors, and you'd have one of the other ones. And you'd give me a brief report next time we have class on what each one of those does. Because class, every one of those proteins is important to this process. That's my point. You don't have to remember all this. What I want you to remember is the overall picture. What we're seeing here. Keep going. All right, as this process continues, stop right there. That's ribonuclease, which separates and starts riding along the DNA. And you see this yellow strand? This is the messenger RNA being built down inside here as it moves along. And as it moves along, it's opening up the DNA, reading the information, and then proceeding. Okay? So go. Stop right there. You can see this is the DNA going along here. This is the protein glob. This is the messenger RNA being produced and released out into the cytoplasm. Lots of stuff going on in here. You watch as we go along. There's all kinds of little stuff floating around in here. Those are pieces of material, including protein, that are needed to go on inside here to produce the protein. Okay? Go. What we're going to do now is blow up this, magnify it, so we can look down inside. Let's stop right there. All right, do you see all these little things coming in? They float in here and they go down inside that protein glob, and there's lots going on down inside there, which we're going to see in just a second. But you can see the DNA out of both sides, but what's happening in here is where the action is. So let's go. Now you're in there. Can you see how that's unzipped? Stop right there. This thing has unzipped by the means of a protein. Thank you. And can you see the code along here? G, G, T, A, A, G, and so on. 3-letter words that came off of the DNA, you remember? That is the DNA. And so what you are doing, exposing this language made up of 3-letter words. And class, if there's a T over here, then the copy is going to have an A, because A and T always go together. If you have an A over here, you would think you'd have a T over here, but you don't. You get a uracil, and don't ask me about that. That's even more complicated. But thymine is replaced by uracil in RNA. There's a whole study about that there are books written about, so don't ask me. But it's a fact. If there's an A over here, you get a U over there, and an A gets a U, a G gets a, what do you think, class? C. G and C always go together. So if there's a C over here, then you get a G over here. So it's an exact mirror image copy of the information contained on the DNA. And they're copied one at a time, class. One at a time. Class, did you know that an average protein has 1,000 amino acids in a row, each one of which has 3 letters? May I say that again? An average protein has at least 1,000 amino acids. Each one of us has 3 letters. And every one of those letters is copied one at a time. So you could have 3,000 letters that have to be copied to make one single protein. Does that make you tired? The problem with videos that I have looked at before is they do not show you how rapidly this takes place. Drew Barry fixes that. So first, just watch. We're going to see it in slow motion, and then he's going to speed it up to how fast it really happens. Are you with me? We're copying every one of those letters one at a time. So you get a whole string of amino acids coded, which makes a
protein.
Right. Watch it. See, one is copied, next is copied, next is copied. That's way too slow. Move it. That's better. You see that? Stop. That's how fast this thing happens. You wonder why you get tired sitting there listening to me? You have 3 trillion cells in your body at least. Every one of those cells is building proteins at that speed while you sit there. You wonder why I get tired? Because I'm working them harder. And that's what's happening. And that's the speed at which it's happening. You can build thousands of proteins in seconds. And by the way, can I throw this in free of charge? Sometimes it makes a mistake. Can you imagine? Did you know built into this process, there's a correction process to correct because you don't want messed up DNA. That'll only change who you are and what you can and can't do. So there is a repair process built in we're not even talking about. Move on. There's the RNA coming out the top, being built at an enormous rate until it's finally finished. Stop right there just a second. I forgot to tell you in that code, not only is there a code for all 20 amino acids, there's a code that says start reading and another code that says stop reading. So when it goes through that ribosome, it says start reading and it reads 3-letter word, 3-letter word, 3-letter words, and then it says stop reading, and between start and stop there's a string of amino acids which is a code for the
proteins.
That's what happens. So this thing is going to keep going until it gets to a stop codon, and then it's going to let it go. Got it? So let's go. Now it's just been released. Stop right there. This is the messenger RNA that's migrated. The one flaw in Mr. Berry's video is he doesn't show you about the migration very well. It migrates up to that hole. And remember, what did I tell you guards the hole? A protein that allows this messenger RNA to come out, and then it closes back up. It's a specialized protein that allows that to happen. All right, so that's already been done, and now we're outside. Let's go on. And it comes out here, and it is picked up by a roving ribosome. Boom, stop right there. You have thousands of ribosomes in every cell in your body, class, made up of
Protein.
and ribosomal RNA and some other stuff. And they're floating around constantly looking for jobs. And this is one of the jobs. It's building a protein right here. So he grabs on the messenger RNA and starts reading. Keep going. Stop right there. Whoops, go ahead. Let's go a little further. All right, you see stuff happening here. You see the codes being read as it reads through the ribosome here. This is the messenger RNA. Oh, this is the transfer RNAs. Watch these guys. They look a little bit like seahorses, don't you think? And they're bringing up the amino acids. And in just a second, we're going to stop. Okay, here's your transfer RNA. This is the specific transfer RNA for that amino acid. Everybody's eyes up here. It's going to show you that there are different amino acids and the transfer RNA for each one is unique. So we're going to watch here. Go. There's a different one, different one, different one, different one, different one, different one. That's enough. Stop right there. Every amino acid has its own transfer RNA, which brings it up to this area here, and it reads this 3-letter word, and it says, get me phenylalanine, and the transfer RNA brings it in and connects it. Now let's go. We're going to look down inside. All right, now we're down inside. Stop right there. See, here's the messenger RNA. The code is right here. This transfer RNA has a code that is the exact mirror image of that code. And up here at this end is the specific amino acid you need to add to this chain. You see the amino acids up here, the code's down here. The code matches the message from the messenger RNA, and you get the amino acid added to the chain. And the problem with this video as of right now is it's going way too slow. So let's watch. So there it comes in one at a time. Stop just a minute. See, when this guy's finished, he's dismissed, and he goes back into the cytoplasm and is recycled. When this one's finished, he goes back and recycles, and so on. And I told you, didn't I, that for every transfer RNA, there's a specific
protein
to go with it to make all this happen. So there's a lot going on here, class. Keep watching, because he's got to make this go faster. Come on, shape up. That's a little bit better. Move it. See the protein being built up there at the top? Here it is coming out the top of the ribosome. Still too slow. Stop right there. I just had to wait and let you watch that a little while. Class, this is unbelievable. Henry Ford never built a factory that touches top side or bottom what this does every microsecond. And there you have your completed protein. No, you do not. That is not finished. A protein is not just a string of amino acids. It's a string of amino acids that gives a secondary, a tertiary shape. And if it isn't shaped precisely correctly, it does not work. And you know what your body does to proteins that don't work? It destroys them. It becomes garbage. So this protein isn't finished, even though it's at its secondary shape. It goes into another system, in some cases called the Golgi body, made of, what do you think, class? And it goes inside there, and there's another, what do you think, class? To help shape it into its final form before it's released into the cytoplasm to do the job for which it was designed. And did you know there are biochemistry texts that read just exactly like that? And then they proceed to spend 10 pages showing you that it wasn't designed. Class, this was designed. Any reasonable person, including Dr. Crick, would have to say, this did not come about by small-scale changes over millions of years, one step at a time, till it finally produced this. No, class, you had to have it all or it doesn't work. Let's go on. Are we stuck? Maybe I need to get my clicker, because we're through with the video. Here we go. Here we go. The chemistry of building proteins in every living cell is sufficient to proclaim there must be a great designer. Ladies and gentlemen of the jury, I would present my case to you and I would say, case closed. I rest my case. Will you convict that there's a designer here? That's what you need to answer if you're an atheist. But I'm not finished. So what happened here? Okay. All right. Because he doesn't need that anymore, because we're through with that. This cell, class, we haven't touched top side or bottom of what's going on in here. There's a whole lot more going on. Because every cell is a self-replicating nanoscale robot. Why do you say that? It's self-replicating because it can and does reproduce itself. Did you know your cells can reproduce everything in them? And they do so. Second, that's nanoscale because it's quite small. Well, that's an understatement. It's 1/1000th of a meter. That's the shape you're going in. I mean, that's how small it is. And then it's a robot because its activities are carried out unconsciously and automatically by precision molecular machines that follow ordinary physical laws. What we've discovered, class, in the last 20 years is there are thousands of little machines working inside every cell in your body. And every one of those machines is more complex than anything man has ever built. And people still say, isn't it interesting what Mother Nature did? Mother Nature must have been something else. How about God? So this afternoon, we're going to talk about molecular machines. What's a molecular machine? It's a natural device on a molecular scale with few exceptions made of what, class? Proteins, able to convert chemical energy to produce linear or rotary motion as well as controlling many biological functions. These little guys are busy doing all kinds of things that if they weren't, you'd be dead. And we're going to take a look at some of those this afternoon. Don't miss this afternoon. I close with this morning. Anthony Flew, a professor of philosophy, atheist, author, and debater for 80 years. He was one of the foremost spokesmen for the atheist community. In 2007, when he turned 80 years old, he wrote this book, There Is a God. How the world's most notorious atheist changed his mind. I just turned 80 in June, class. I hope I'm open-minded enough at 80 to still change my mind if the evidence is overwhelming. And I hope you are too if you're listening to this and you're an atheist or an agnostic. Please examine the evidence. It's overwhelming. I'm not touching the hem of the garment here. Here's what Dr. Anthony Flew said about himself. It has become inordinately difficult even to begin to think about constructing a naturalistic theory, that's a theory without God, of the evolution of that first reproducing organism. The enormous complexity by which the results were achieved looks to me like the work of intelligence. That's an honest man talking. It now seems to me that the findings of more than 50 years of DNA research have provided materials for a new and enormously powerful argument to design. Is that what I'm doing? I hope so. I'm trying to show you, class, the evidence for design has never been so overwhelmingly powerful as it is today. And please believe me, one of the things I'm going to do when I go home tonight is I'm going to get on my knees and ask God for forgiveness. Because I haven't done living things their justice. They're way more complicated than I have told you. Is it reasonable to believe in God in this scientific age? Class, it's more reasonable than it's ever been in the history of mankind. And foolish people are telling you, you belong in the dark ages if you believe in God. It's the exact opposite. You belong in the dark ages if you don't believe in God. So come back at 1:30 and let's study molecular machines. It's fun. And there's 2 videos. I hope you can come back and I'm done. Thank you, Troy. God bless you.